Welcome everyone for participating today and for joining our session. My name is Rob Murphy. I'm the Executive Director of the Institute for Global Health and Director of the Center for Communicable Diseases, Global Communicable Diseases here at the Feinberg School of Medicine. Thanks for joining us this afternoon in our latest installment of the Institute's First Friday Seminar Series. Today we have a very exciting presentation on COVID-19, the global epidemiology and response. Our panelists today, we have three panelists, include Dr. Chad Achenbach, who's Associate Professor in the Division of Infectious Diseases and Preventive Medicine here at Northwestern. His research focuses on ways to improve treatment and outcomes of patients with HIV antiretroviral therapy, and in particular, his interest lies in molecular markers of aging among the HIV population and epidemiology of HIV-associated conditions, such as cancer and cardiovascular disease. Dr. Karla Satchell is also with us. She's a professor of microbiology and immunology and director of the Center for Structural Genomics of Infectious Diseases here at Northwestern. Her research focuses on the role of secreted protein toxins on bacterial pathogenesis. Here at Northwestern, she's the lead investigator on a project which is looking at structural components of the coronavirus to ultimately understand how to stop it from replicating in human cells through a medication or vaccine. And our final participant is Dr. Mike Ison. He's a professor in the Division of Infectious Disease and Surgery in the Division of Transplant Surgery at Northwestern. His primary clinical and research area focuses on infections and transplant recipients, and his research is further focused on viral infections, including norovirus, CMV, respiratory virus infections like influenza and adenovirus, particularly as they occur in transplant patients. He's the chair of the Adaptive COVID-19 clinical trial through NIAID and DMID at the NIH. There will be time for questions at the end of this presentation, in which you are able to submit your questions through the Q&A feature at the bottom right-hand corner of your screen. Portions of this webinar will be featured in an upcoming episode of Feinberg's Breakthrough Podcast, and you can actually get CME credit for listening to that podcast episode, which will be available later this week. Dr. Achenbach, I'm going to turn the session over to you, which you're going to chair the rest of it. Thank you.
Okay, thank you so much, Rob. And today we are going to go through this following plan for our webinar. I'm going to start off and give the big picture overview of the current global epidemiology and public health response, essentially, to COVID-19. And then Dr. Ison will talk about some specific clinical aspects and current therapies and what we're learning from around the world in terms of what we can do for this. And then Dr. Satchell will talk a little more nuanced about SARS-CoV structure and novel therapies and maybe ways we can look at the structure to come up with vaccines and therapies. Then we'll have a Q&A session. So we achieved a big milestone yesterday. We have now over 1 million cases of COVID-19 in 181 countries globally. We're leading the way here in the United States with 245,000. Italy and Spain are next with about half that many, just over 100,000. I want to point out here that Germany actually is the latest country to leap above China now with around 85,000 cases. And we're still seeing quite a big burden of this problem in France, Iran, UK, and then some newer countries that are getting above the 10,000 point are Switzerland in more Central Europe and then Turkey. So in terms of death, it tracks pretty well with where the cases are, but there are certain countries that appear to be bearing the greater burden of the death in Italy and Spain, and then we are catching up at around 6,000 deaths. And then France, China, and Iran are next in order. And then again, some of these smaller European countries are now hitting 1 in 2,000 cases of deaths within their countries. So how did we get here? I think it's good to kind of think back again on the timeline of things. So we noted the first cases in China and really clusters of them in kind of mid to the end of December of 2019. The virus, which at the time was called novel coronavirus 2019, later identified and called SARS-CoV-2, was identified on January 7th. The first death of this globally was on January 11th. And the sequence fairly quickly was done and shared in China on January 12th. And not long after this, the first test kits became available for SARS-CoV-2. The first case outside of China landed in the middle of January, and then about a month after that is when we really started seeing more spread to other WHO regions of the world, with spread to Iran, Italy, and South Korea in the middle of February. Our first death here in the United States was the end of February on the 28th. And what we're seeing here is really kind of three phases of logarithmic growth is the way I like to think of it. So to get to that first 10,000 cases took about 31 days. If you look from the end of December all the way forward, then you can see here it took up until throughout the entire month of January to get to 10,000, but really it was about 14 days when we really had good testing and could understand the cases that were coming in China. We pretty quickly got to that 10,000 threshold. Then things slowed down a little bit and leveled off. You can see there was a little bit of a bending in the curve here globally as China got things under control kind of in the middle of February. But then as it popped into Europe and other parts of the world, we started seeing, again, logarithmic growth. So this is on the jump from 10,000 to 100,000 took approximately 35 days. And we're now on a little bit quicker pace. To get to 1 million cases took us under 30 days now, approximately 28 days to get to that milestone. I think it's good to take a look at the epi curves of China and what happened there, because they've kind of been through the whole first wave of this at this point. And so I think some things to note here in China is the blue bars were the laboratory confirmed cases in Hubei, and then the rest of China are in green. They had about a 2-month epi curve here over the span of the whole thing. They had a full lockdown of the entire city of Wuhan, and it took about 14 days before they hit their peak cases in that region, in the Hubei province. I think one thing to note here is the rest of the country tracked pretty closely, showing how quickly this spread from outside of Hubei into other parts of China, but also how in other areas and other parts of China, they're able to get this under some semblance of control fairly quickly. Unlike, I think, what's happening now in the United States, where it's kind of moving from large urban center to large urban center. They kind of, here in China, they saw one kind of large spread to other parts of China, and then not a lot of jumps or bumps within their case counts over time. I think they had the benefit, though, of knowing they had kind of a point source in some ways. They knew that there was a source of this infection within that one province, and they could center all of their and concentrate all of their epidemiologic measures around that one place. And so that was something they were able to use to their advantage. So what is going on in China? Do they still have cases and are people dying? Well, as of yesterday, there still were 100 cases in China and 6 deaths. But when you look at those curves more carefully to get a sense of what we're in for, they had approximately 21 days or about 3 weeks where they were at their, what I would call kind of their cap of cases, where they were kind of in this realm of 1,000 to 3,000 cases per day. That's about how long that went on for. And their most was approximately 5,000 in one day. And it was about a similar period of time, actually, about 3 weeks in which they were seeing over 50 deaths per day in their kind of cap of cases in their curve. And the most that they had was 250 in one day. So what are the current areas that we were seeing the most spread? So these are the areas I think are our current hotspots, mainly in Europe and here in the United States. And so what we have here in this graphic is the logarithmic growth, essentially. So it's the log scale on one side of cases, and then the days since there are 100 total confirmed case. So if you at any time point, if you draw a line, a tangent line, and then you look at the slope of that, it can give you an idea of how quickly this is moving in terms of cases. And then I'll show, and we'll talk about deaths after this. And you can see that we are still on a pretty steep linear curve of this. But other countries such as Spain and Italy, so we're in green here, and Spain and Italy and France are in blue, the European countries. There is some bending here. There's some movement within that linear tangent line to flatten that slope. So there has been a decrease in cases in those places. Germany has had some acceleration in cases, largely, I think, due to testing. And then we're still seeing cases at a pretty fast rate, especially in Turkey and other parts of the world like Iran. What about the highest death rates? They don't track perfectly. They correlate pretty well, but the deaths in some places are lower than others. So, and I think this is a reflection of areas of the world where there's more testing available and they're doing more testing per capita. They're able to detect more mild cases, and so their death rates may be slower. It definitely appeared that way with Germany at first, but they appear to be catching up somewhat in the slope of the curve for Germany here. The tangent line there is increasing over time, I think. And I think what this shows is this difference between the ability to detect cases. So is there, are we unable to detect cases? And then also we need to think about the ability to detect death as well. So misclassification of either diagnosis or death can be challenging. In a lot of settings, such as the US and Europe, where many of the deaths are happening in the hospital, I think we have a pretty good idea of the deaths and what we're capturing. But in other parts of the world, such as Iran and Turkey, their death rates seem to be low, but the anecdotal reports from the field are that there are a lot of people in the community and at home who are dying at home and dying in the field. And I think that this is something that we might see more as in other countries as well. And so when looking at these numbers, you kind of have to balance what is the healthcare system like that we're looking at here in terms of their ability to be able to correctly ascertain diagnosis based on testing, and then also death based on the way that deaths are calculated within a population. And so this gets at that point of the testing and the total tests that have been done. We have accelerated our testing quite a bit here in the United States. There's also a lot of testing going on in Germany, Italy, and South Korea, and this has increased over time. We were a little bit behind on the testing, as I think we all know, and have been catching up. But when you look at this per capita, it gives you a better sense of how well we're capturing the entire population and maybe some more mild cases within our testing. And you can see here, leading the way is Germany, and we drop here down to fourth in terms of testing per capita. And South Korea is another country where they had a very massive early on testing campaign, were able to detect more mild than asymptomatic cases, and that because of that, they have lower death rates as well. Italy, on the other hand, they do appear to be doing a lot of testing per capita, and despite this, still have a higher death rate, which again, as I mentioned, is leveling off, but still appears to be a problem there. And I think the testing issue is illustrated with even just some of our local data here at Northwestern Medicine. So where we were on a pretty steep curve of our positive testing here, you can see in these gray bars, up until March 26th, where we had outpatient testing, we had a lab and drive-through testing, we had a lot of testing available. But then we realized that we were having some issues potentially with supplies and had to decrease the test availability only to emergency rooms and hospitals. And I think a lot of places around the United States and other parts of the world are experiencing this issue, where we would like to test everyone, outpatients and more mild disease, but when it comes down to it, with some of the supply issues and whatnot, we maybe are limited in what we can do. And you can see there's a drop off here in the number of positive tests. So just because the epi curves may level off or drop doesn't mean that it's all because of actually getting things under control. So we need to be careful with those numbers. So what are the next hotspots? I think South America, Africa, and Central Asia are areas that are growing. So we're seeing quite a bit of growth here with now cases above 5,000 in Brazil, 3,000 in Chile, here in the red line, and then Pakistan and India are growing as well. We have now over 1,000. The only country over 1,000 in Africa right now is South Africa, and we're seeing pretty low numbers in many of the places that our institute works around the world, such as Nigeria, well, it's only 151, and in Ethiopia and Botswana as well, having fairly low numbers. Although there's some concern that they don't really have the testing capabilities, so how do we truly know? And so when thinking about this next phase and this next wave of hotspots, I think there's some concern, mainly because of poor health and public health and medical infrastructure. There's a lot of poverty and care disparities, crowding in these countries, and the overall life expectancy is lower. And the confounding fact of co-infections, such as tuberculosis and HIV. But I mean, I think a few of the things that might help them are at least in Africa, potentially a bit younger population. We know that at least the more severe forms of COVID-19 are in older populations that Dr. Ison will talk more about. And they've also dealt with many prior infectious disease crises. Here on the slide, you can see them dealing with Ebola. We also have had large-scale public health emergencies and issues with Zika in South America and Brazil. And then I think what this brings in is how important it is to have collaborations with global partners, both within institutions like Northwestern, but then within countries as well. And many of these countries around the world have a lot of collaborations with the United States and Europe, and hopefully we can impart our knowledge onto them as they start to face this. So what can we do without a vaccine or treatment? So this is very much geography and time dependent, depending on where you are, but I like this graphic they have here from Thomas Peo, who's a businessman and engineer who's done a lot of writing on this. And there's kind of the strategy: if we do nothing, that's not an option. If we mitigate things, we can slow it down. But really, we need this hammer approach where we think of some of the more extreme public health measures. So we definitely need to be doing containment issues, case finding, isolation, quarantine, and then mitigation, things like closing schools and universities and social distancing. But then to bring in the hammer, we really need some more extreme measures: stay-at-home, mandatory quarantine, closing businesses, and deciding whether or not to do that with essential or non-essential businesses, and then mandatory quarantines, making people stay home, government takeover, eventually stopping travel and martial law. So these are all things within the armamentarium of things we can do to slow this down. Social distancing, I think we know that this can work. This is in the Spanish flu in 1918. We know that with the cities that did better jobs of social distancing, were at least able to make this into a two-wave epidemic and blunt that initial wave of high mortality. They relate to the game in Philadelphia, and we saw this high spike, whereas it was spread out over time, the mortality in St. Louis. And I think the idea here is an individual can make a difference, and behaviors do make a difference. So if one person transmits it to three, if our transmissibility is from 1 to 3 people, then one of those people decides to work from home and doesn't infect anyone, and then it passes on from there, and then one person decides not to go to some barbecue, you can see how this can really shut down the spread very quickly. And even with just a few people distancing themselves and not transmitting to 2 or 3 others, we can cut this epidemic down very quickly in half or even in a quarter. And then when thinking about it, timing is important too, how long we do the social distancing. This is just some modeling exercises looking at doing it for 14 days versus 2 months. And when we extend it out to 2 months, you can really squash that curve, flatten it down, and bend things in the right direction to spread things over time and reduce the total number as well. So some good examples from other places around the world are Singapore, Taiwan, and South Korea. What did they do? As you can see here in the green line, South Korea, they started seeing linear growth in a high number of cases, but we were really able to shut it down and bring it down. And then the examples of Taiwan and Singapore were essentially it never really gained a big foothold. They never had this explosive growth like we've had in other countries in the world. So one thing to note is these countries are a little bit smaller, and they have a very central coordinated public health infrastructure that was able to coordinate a massive public health response on a large scale in many parts of the country. And they learned from their prior experience with the first SARS-CoV, and I think had some fear because of that and seeing how badly that decimated both their economies and their public and health systems. So they had very aggressive detection, testing, and isolation early on. They also planned for surge capacity supplies. They provided money to healthcare workers, called for volunteers, and brought them into the places that was needed. They did a lot of social distancing and home quarantine, things that we're doing now. They provided economic assistance to their citizens, and then travel bans. They all had entry restrictions, and then other bans of gatherings was actually only done in South Korea, over 250. But one thing I do want to point out is what they did not do. None of these countries actually closed any public schools. They did close some private schools. They did not close any non-essential businesses, and they never closed down entirely any cities or borders. The only city to date that's been fully closed down to control this was Wuhan, China. So a few anecdotes I think that are important to think about that have been coming out lately that help, I think, us as individuals is There's been reports just yesterday or a couple of days ago of pre-symptomatic clusters in Singapore, I think 7 different clusters that led to cases where they could clearly document through CCTV and other means that there was clear asymptomatic or pre-symptomatic transmission. Those people eventually did go on to have symptoms, but at the time they were asymptomatic. And then there's been some more clusters of asymptomatic spread in China. So this helps us understand that we really do need to be doing social distancing, home quarantine. You can't predict who's going to become symptomatic in the next 2 days, right? And then this is where masking is potentially important as well. When there is a breakdown in that social distancing, which can be unpredictable, you have another layer of protection. There's been clusters of church groups. What do people like to do at church? They talk and sing, right? So this shows the importance of how respiratory droplets and how masks can be helpful in reducing the spread, especially when in closer quarters with people. And then all of the outbreaks on cruise ships, jails, and nursing homes. I mean, these are clues to us epidemiologically that surfaces are key, and decontamination of surfaces are going to be a very important part of this. And my prediction is that when this is all over and we look at and we understand the transmission better, we'll see that surfaces are a big part of it, and hand washing and decontamination of the environment will be key. One thing to think about is how does this compare to other pandemics? So we did go through a pandemic back in 2009 with the H1N1 pandemic, and I think it's good to reflect back on that. There's been a lot of talk about these prior pandemics and how does COVID compare. Well, I think it's pretty clear now that we're off the charts way above where we're going to be with the 2009 H1N1 pandemic. Remember, that's influenza. We had therapeutics for that. We were able to quickly make a vaccine within 6 months because we had experience with flu vaccines. And so it was a very different situation. But just thinking about the raw numbers, here's the epi curve here. There were essentially 2 waves of this, and this virus is still with us today. As you know, it's a part of our quadrivalent vaccine is the antigenic drift that happens each year within this virus. But even at its peak back in the end of 2009, we were seeing 35,000 to 40,000 per week of this virus, whereas right now we're at 77,000 per day. That's where we're at this moment, and that's not even peaking yet. It was estimated after it was all said and done that there were approximately 284,000 global deaths from this. We're at 53,000 in only a 3-month period of time. And that global death estimate was over after about a year of that outbreak. And that was 12,000 deaths in the US. We're already halfway there after only 5 weeks since our first death was reported. So we're wildly accelerating beyond where we'll be with that pandemic. So what can we expect in the US here coming up? Well, this is some modeling exercises that have been done from the Institute of Health Metrics and Evaluation in Seattle. And I think this has been in the press a lot this past week, and trying to think about what's coming and what we can expect here. And one thing I want to note with all this modeling is it's all pretty much based on Wuhan data. So again, they give it more point source where it's spread out from one place. And it may be more challenging here in the US where it probably was introduced to many places all at once. And so how we respond could be a little bit different. But in the modeling estimates also assume that we have a complete social distancing through the end of May, and that there's some level of social distancing that goes on throughout this whole observation period, which is essentially till August. But all that being said, these estimates are that we're going to reach our peak in daily deaths in about 2 weeks at around 2,600. And I drew this in here. I think this is kind of more how it will look. There's going to be ups and downs. There's going to be different cities that have death waves and different places epidemiologically that will come in. And the whole curve may be smoothed out to look more like what's in the model. But as you can see, starting off, it's already a jagged line. I think what we need to be thinking about is what is the range of possibility. So how long might we be at a level of 1,000 cases or more? You can see here, it could be as early as getting below that level kind of in the 3rd week of April, or it could go as far as the 3rd week of May. So we have about there's a lot of variability in these models, and there's a lot of uncertainty of about a month in the timing, and also in the peak. Is the peak going to be at 1,500 cases per day, or is it going to be closer to 4,500? So time will tell. We'll see where we end up, and I think a lot of these models possibly could be underestimating things. In terms of death, it's based on these models that we get to this estimate of around 100,000 deaths by August. But again, there's a lot of variability here from anywhere from 40,000 up to 180,000. So what can we do going forward? So I think the best way forward is us as individuals, social distancing, stay at home. I think we can wear masks. It's a new recommendation now. I do think that's important, and here's why. We try our best to do the social distancing. That's the first thing we can do. But there are situations, going to the grocery store, even when you're out walking on the street, where sometimes people come close to you or come up to you and you can't control it, and that's out of your control. And that does provide another barrier when that does happen, or in situations where it's really hard to keep that 6-foot distance, especially healthcare workers in hospitals. We do have a universal masking policy at Northwestern now, or if you're out at a store or somewhere where it's a little bit more cramped space, I think that's just another barrier of protection for you. In academia, we need to keep schools and universities closed. The government can help support people economically, and also vulnerable populations with social services and economic stimulus. Our medical systems, we need to ramp up supplies and capacity. We need to think about, is there other facilities we can use as isolation centers or hospitals? Hasn't been a lot of thought about this, but can hotels or other places be turned into extensions of our medical system? And in terms of public health and medicine, what are ways we can optimize technology, more point of care molecular diagnostics? We will be getting into that here at Northwestern, where we're going to have more rapid and somewhat more point of care diagnostic testing capability, up to potentially even 2,000 tests per day. That's going to be rolling out here within the next week. Also, we need to think about real-time monitoring. Are there novel ways we can think about electronic monitoring people without infringing upon their civil liberties and privacy through apps or through video monitoring, whatever it might be? And then, of course, drug and vaccine development. And then I think we need to be thinking about, as this evolves in the United States, do we need to stop all transport and air travel in and out of cities? Does this extreme measure need to be, does that need to happen? Do we need to go to that extreme at this point? We may not need to, but we need to be thinking about, is that something we need to do? Closing cities down like they did in Wuhan, and closing all transport in and out of cities, as has been done if it gets more severe. But I think if we do, one thing we do know is the speed of which we act. If we do it more forcefully and do it quickly, we can kind of shorten this problem and get things under control, and then get back to our usual business as soon as we can. So with that, I'm going to turn the next part of the presentation over to Michael Ison. So he's going to talk a little bit more about the clinical aspects and what we're learning from other parts of the world in terms of that and therapeutics.
Perfect. Well, thank you all for joining today. I'm going to try to move through this relatively quickly. Here are my disclosures. What I hope to talk about a little bit about is the virology, transmission, clinical course and prediction of outcomes, and then briefly about some management options. So what we know is that there's a range of clinical disease, and while the pyramid shows a relative proportion, I would say I'm not sure that we know what proportion of patients have asymptomatic disease, since we haven't been doing great studies of a whole population to understand that. Looking at some data coming from countries such as Iceland and South Korea, there probably is a larger pool of patients with mild or more symptomatic, minimally symptomatic disease. Where most of our data comes from are the patients that have severe disease requiring hospitalization or ICU level care. In terms of epidemiology, what we know is that from infection to development of symptoms, it's somewhere in the neighborhood of 3 to 5 days. Once the patient becomes ill, they typically have a day or two before they start to seek care. And then hospitalization occurred on average day 9 after symptom onset. Once in the hospital, it takes about another day before they get more severe respiratory distress and require ICU level care. So on average, day 7 to 9 to admission, day 9 to 10 severe symptoms with progression to ICU level care in those patients that progress, which thankfully is still a subset of the patients that get ill. From the data in China, what we know is that patients that are older and that have underlying medical conditions are more likely to require hospitalization and develop severe illness. I think it's important to recognize that the two most common underlying conditions that were seen were diabetes and hypertension, Again, very common problems in the United States and elsewhere around the world. Overall, severe infection requiring ICU level care or death accounted for 6.1% of the patients in China. 5% required ICU level admission. About half of those patients required mechanical ventilation, and mortality in China was about 1.1%. In terms of clinical presentation, many but not all patients will have fever, cough, and fatigue. There appears to be some, for many patients, a biphasic illness where they have more milder symptoms with a second wave where fevers really ramp up. They start developing shortness of breath and get progressively ill. Signals of patients that are more likely to progress to require hospitalization or ICU level care are patients that have exceptionally high fevers, typically above 102, 103, when they have hemoptysis or blood in their sputum when they cough, patients that have low white counts, especially low lymphocyte counts, and evidence of end organ failure. This graphic shows one of the first cases seen in the United States where you can kind of see what symptoms happened at what time. Unlike the common cold, rhinorrhea is oftentimes not an early sign. Cough is usually present throughout much of the illness, again, with peak fevers developing several days after onset of symptoms. Fatigue is relatively persistent as well, with intermittent GI symptoms in some patients. In terms of diagnostics, one of the earliest studies, and this has been shown in future studies, including ones coming out of Italy, is that chest imaging, particularly chest CTs, sometimes give you a much earlier signal that a patient that has mild symptoms may have COVID-19. Typically, what's seen is shown in the CT scan, bilateral ground glass infiltrates suggesting pneumonia that's scattered involving both sides of the lungs and often all lung fields. It typically starts in the lower airways and progresses upward. Mortality, as I had mentioned, is really consistently higher in the older population. Overall case fatality has been described to be a little bit higher in Italy, and we are seeing, at least in the United States, that there's a higher number of younger adults that are being admitted to the hospital in the ICU. Nonetheless, as you get older, decile by decile mortality increases such that patients in their 70s have a case fatality somewhere in the 8 to 10% range, and those that are over 80 really have a much higher mortality, 15 to 20%, depending on the population that you're looking at. In terms of biomarkers, there's been some effort to identify potential biomarkers suggesting more progressive illness, although a number of markers seem to be associated with patients that have progressive illness requiring ICU level care and death, as shown in the red lines here. The only one that was statistically different was D-dimer. And I'll say from my experience of seeing some of these patients, the patients that tended to have relatively stable, low-level D-dimers tended to do well and get discharged, whereas those that had rising D-dimers were more likely to get admitted to the ICU. There are also some signals that cardiac biomarkers may be predictive of patients that are more likely to develop severe disease, including cardiac complications, including troponin T and BNP as markers of cardiac dysfunction. There's a lot of questions about asymptomatic shedding and the risk that asymptomatic shedders result in transmission. There's been some modeling work that suggests that asymptomatic shedding likely accounts for about a day worth of transmissions that aren't accounted through other known contacts for the patient population. We're finally starting to get some information on virology. When I pulled this graphic out, it was on MedRxiv. It has now been published in Nature as of yesterday. And basically what you can see is patients have the highest PCR positivity relatively early on after symptom onset. And when patients have high viral loads, not surprisingly, they have detection of culturable virus. By culture, at least, detection wanes by day 10 to 11 in patients, most of whom required hospitalization. And this data is coming from Germany. There's clear evidence that patients that have more severe disease are more likely to have higher viral loads, and that the viral loads tend to persist for a longer period of time, particularly in those patients that require ICU level care. Turning lastly to management options, I think the most important thing to say is that there is not a single drug to date that has been proven to be clinically effective in treating or preventing the SARS-CoV infection or the outcomes of COVID-19. There are a couple of things that have been studied. Early signals and consistent with studies with SARS and MERS are that steroid use, especially very high-dose steroids, is associated with prolonged shedding and no significant improvement in survival. There have been a number of agents, including convalescent plasma, Lopinavir-Ritonavir, chloroquine and hydroxychloroquine, IL-6 blockers, and remdesivir that are currently undergoing clinical trials. There's a huge laundry list of other agents, and literally every day I get approached about other potential therapies that could be used. I think the upshot of all of this is we really need randomized clinical trials to really inform our practice. As someone who's done research in influenza for years, I'm shocked that so many people are jumping on therapies that don't have clear proven benefit, when just a few months ago, when we were in the middle of the flu outbreak, people were shunning the use of drugs that have been proven through randomized trials to be beneficial. One of the first studies that came out and was published a couple weeks ago in the New England Journal of Medicine was this trial of Lopinavir-Ritonavir, in which they gave patients that were PCR positive and had either hypoxia and evidence of lower tract disease, Lopinavir-Ritonavir twice daily for 14 days, or placebo. Primary endpoint was time to clinical improvement, and then a number of secondary outcomes followed. When we reviewed this for our journal club here in the ID division, I think the main thing that I pointed out is that although it wasn't the primary endpoint, looking at virology, which is on your left side of the graph here, there was really no benefit in terms of more rapid clearance of virus with the use of Lopinavir-Ritonavir. And so whether this means it's not having a true antiviral effect in humans or just not getting to the site of infection is something that needs to be studied better through the samples from this. But combining that with the failure to show a clinical benefit in the primary outcome, I think most would agree Lopinavir-Ritonavir is not a drug that we should be thinking about to pursue further. There's been a lot of enthusiasm for chloroquine and hydroxychloroquine because of theoretical benefits in terms of blocking or interfering with the ACE2 inhibitor, acidification of the endosome, interfering with trafficking of the virus into the cells, and then potentially immunosuppression, which may be either a benefit or a detriment. Theoretically, you can achieve therapeutic levels with this medication. And there's been a lot of enthusiasm driven in part by this study that now has been published, which included a relatively small number of patients treated with hydroxychloroquine. Some also got azithromycin if they had evidence of underlying infection and required therapy. But I think we all should read the article very carefully, because despite this, what seems to be a clear benefit, they didn't include 6 hydroxychloroquine patients that were lost in their assessment because 3 went to the ICU and 1 died, 4 of which I don't think we would consider as successes. 1 was discharged and another stopped the medication because of nausea. A subsequent study, again, very small numbers, recently came out of China, but again, we need a large randomized trial to understand the efficacy of this drug. IL-6 receptors are of clear interest to many because it may impact the cytokine storm. The optimal time of using these interventions and the appropriate patients to use these in still require some studies. Thankfully, there's several randomized studies ongoing that should help give us an answer. In terms of antiviral agents, remdesivir is a drug that is an RNA-dependent polymerase inhibitor that is active against a number of RNA viruses. including SARS-CoV-2. The EC50 is in a therapeutic range. There's currently an ongoing randomized trial of this medication that should help us understand the benefit. Lastly, I think there's now a bloom of enthusiasm for the use of convalescent plasma. The important thing to recognize is you need recovered patients to actually collect this plasma from, and so this will be something that I think we can study in the United States in the very near future. Looking at the first study, so 5 patients that were treated in China with convalescent plasma, they appeared to have some degree of clinical improvement. They frequently had other therapies provided. The important thing, which isn't shown here, is that the intervention was very effective in boosting antibodies to SARS-CoV in the blood of the patients that received this. There appeared to be both a clinical and virologic benefit for these patients. So with that, I appreciate the opportunity to present to you today, and I'll turn it over to Karla.
What am I not doing? Oh. Is my camera on? Can you hear me?
Yep.
Okay.
Yes, we can hear you and see your slides.
Oh, okay. All right, so I'm going to be presenting today about the role that we're doing here at Northwestern and our consortium, looking at structural biology in hopes of actually improving drug discovery for SARS-specific interventions in the future. Okay, so I'm actually the co-director of the Center for Structural Genomics of Infectious Diseases. This is a large multi-site center that's headquartered here at Northwestern University. And the work I'm going to be talking about today is from this consortium, including, I'll talk about data from the University of Chicago, as well as Purdue University, and with assistance from the UC Riverside. We're an NIAID-funded center, and we've been funded since 2007. So the coronavirus, and from a structural standpoint, is a virus that we see here under electron microscopy, and we see this envelope virus with these large spikes come out. These spikes appear to be a crown, and so this crown is where we get this name coronavirus from. We just look at the actual structure of this virus. We see that it's actually an elegant virus, but it's actually made up of only 4 proteins. We have the envelope protein and the membrane protein, which form the circle of the actual capsid. And extending out from this is this spike protein, which is a glycoprotein that extends out. Within that capsid, you have the nucleocapsid protein, which is the most abundant protein of the virus, and that wraps the single-stranded RNA genome. And these particles are around 50 to 200 nanometers. As we know, just from the talk that we've had from Michael Ison, that this causes this severe disease. This is one of the first x-rays that came out from a 44-year-old man in China that progressed to various disease very quickly. And for patients that are in serious disease, we do need to think about very specific drugs to treat active patients. And so this is where structural biology can have an impact. One of the things that we want to think about in terms of how best to think about new drugs to treat COVID would be, first off, is to repurpose current drugs. And this is somewhat what you've heard just now about from Michael Ison with Remdesivir as well as chloroquine. The advantage of this is it's very fast. These drugs are FDA approved, they're ready to grow and ready to enter into clinical trials. Antibodies, as similar to convalescent serum, can be a medium fast route. You need humans that have had the disease. You then need to isolate those antibodies, determine those that are most effective, and then turn those into new drugs. In slower components, we can actually get into new drug discovery, and this is where our center has been involved, which is really to pick up and reboot SARS research, as well as participate in de novo drug discovery. And this can be a very slow process. It's not something that we're looking at being very effective in the very short term, but it's really important to note if we had not stopped SARS research, we would absolutely have very specific drugs available today. And so it's important that we get started now and hope to have benefit as this disease continues to spread throughout the world. So if we just look at a standard general viral life cycle in this very simple cartoon, you can see that, we first have attachment, and then penetration, and then encoding, and then followed by biosynthesis and assembly and the release of particles from that cell. And the primary areas where you can actually think about developing new drugs would be at this attachment phase, and this is where antibodies can be very protective. Penetration, chloroquine blocks that penetration level, and so that's where the interest in that drug comes from. But this biosynthesis level, after the RNA gets in and you actually need to make new viral particles, can be a very potent place for small molecule drug development. So this is a much more detailed look at the viral life cycle, and I'll take you through just the left-hand side of this. We're going to show you what's going on in terms of how structural biology is impacting drug development against the background of this viral life cycle. So I'm going to start here at the very top of this slide, which is looking at the receptor ACE2 interaction. And so here we actually come back to the spike trimer. So this again is that protein that's actually extending out from the virus that makes this virus appear like a crown. And this protein actually is a trimer, which is 3 molecules that are bound together and exposed on the surface of this virus. And this has 2 portions of it, the S1 and the S2 domains. And it's this S1 trimer that actually interacts with the host cell receptor. And the host cell receptor in this case is ACE2, which is a protein that's expressed on the lungs of humans. And initially there's this steric clash that occurs. So there has to be some wiggling of that spike trimer that then allows it to reorganize and engage a single trimer engages a single receptor. We then have a conformational change as well as a processing event that leads to maturation and fusion of this virus with our membrane that allows that virus to enter. If we look at the cryo-EM structure, this is a cryo-EM structure that was solved by our sister center, which is the other NID-funded structural genomics center present in Seattle. And what they did is they did this beautiful cryo-EM structure of both the closed and this open conformations. So in the closed conformation, you see that there's no extension, and so there has to be that wiggle that allows it to then expose one of those trimers to reach out, and that then binds that ACE2. And what they looked at when they looked at the binding affinity of this compared to how SARS binds, they found that SARS-CoV-2 has a slightly better binding efficiency for the ACE2 receptor. And this isn't due to the fact that it actually binds better, but it actually binds longer. So in biochemistry, we refer to this as the K-on is the same, but the K-off is lower, so it actually binds longer and better. So the other interesting thing about the spike trimer, and that's very unique to SARS-CoV-2, is that it has these extra 4 amino acids in that region. And this is where that cleavage event happens between S1 and S2. And this introduced what's a furin cleavage site. So human cells actually have this furin protease exposed on the surface of our cells. And so what this means is that this virus is sort of pre-activated in order to go into human cells. And it's thought that this is incredibly important for the transmission of this from bats into humans. If we compare this, though, to SARS, regular SARS, we see that it actually didn't lead to dramatic improvement in the ability of the virus to enter, and this was shown in 2 different kinds of cell lines. Interestingly, actually, if they repaired that, they actually saw potentially improved viral entry. So in a human cell, this didn't cause an advantage, but this did cause an advantage of its movement into the human population from bats. So following up on where we are now with regards to drug discovery that's oriented around spike, we now have these beautiful structures, and there's a worldwide effort now from mice as well as from humans to identify antibodies that will be very specific to block this spike ACE2 interaction. This is being done just by looking at virology, but also in a very, very high resolution structural way, similar to that cryo-EM structure I just showed you, looking for human antibodies that are going to bind to that structure so that we can understand where we can best inhibit the interaction. This is also incredibly important for vaccine development because we can understand which portions of that viral particle do we need to be included in the vaccine for long-term protection, for development of long-term protection. So then if we come back to our viral life cycle, now that we've entered the cell, we've gone through a membrane fusion event, the next really important aspect of that viral life cycle is the translation of that RNA that's been delivered inside the cell. And this translation is done by our normal host cell ribosomes, and then that leads to the first translational product. So we can zoom this out in this diagram here, where what we see is the first translational product is ORF1A, and then there's also a second, there's a frame shift mutation that you get a slip strand translation that leads to a much larger transcript called ORF1B. And this single long protein then gets proteolyzed by two of proteases that are encoded within that large protein. And that is the 3CL protease, also called MPRO for main protease, also known as NSP5. And then another protein called PL protease or papain-like protease, also known as NSP3. And this 3CL protease cleaves everywhere you see this light blue triangle. So it cleaves up the protein from itself all the way to the right, so from NSP4 all the way through NSP14, whereas PL protease cleaves between NSP3 and NSP4 and also releases NSP2 and 1. And so these two proteases are the first viral enzymes that are involved in building that viral particle. And because this is the very first step, inhibition of proteolysis by 3CL-Pro and PL-Pro is considered to be the most druggable event in the coronavirus life cycle. And so this is where a large amount of work globally is being directed. And so one of the big studies that's been done was done at the Diamond Light Source in the UK. They did what's called a fragment screen. And what they did is they took the structure that had been determined of MPRO by a group in China, and they took that protein and screened it using high-resolution X-ray crystallography. They screened 1,300 compounds and identified small fragments that would bind stably. And these small fragments can then be used by the research community to assemble and build ideas for new drugs. So all total, they identified 66 active fragments and 44 of which were bound. And this was actually released to the public community on March 17th and March 24th in two separate releases of this study. And so this actually got a lot of press. However, in the CSGID, we actually felt like we were way ahead of what they were doing in the Diamond Light Source. And this is because of work that had been going on at the CSGID by Andy Messaker at our Purdue University site. And here is a structure that we released on March 25th, which is the structure of MPRO. And we see the ribbon diagram drawn from blue at the N terminus through red at the C terminus. But what you see bound into this active site is this compound 77. And compound 77 is a very potent inhibitor of 3CL-Pro. So how did we get to this so quickly? And the reason is because, as I mentioned, there has been SARS research going on all along. And one of the CSGID-funded projects from 2018 to 2020 going on at Purdue was this idea that coronaviruses could reemerge. And so we needed to be doing drug discovery well ahead of its emergence. And so compound 77 came out of a broad screen looking across the coronaviruses at very different, at those that affected pigs and ducks, as well as humans and bats. And so this is now a lead compound that has the potential to move forward. We've released that structure to the community, and as we're going on with, as Andy Messaker's group in Purdue is moving on with advanced drug development. Now, if we look now at this other protease, the PL protease, This is going to follow a very similar line. The APO structure of that was solved and released to the community on April 1st, as well as there's structures that have come out of Andy Messaker from these other animals, and so we see here now that this can now move into that same line of fragment screening as well as ligand screening. And this has actually been, because it's been funded through this project, to look at coronaviruses, ligand screening has already happened and lead compounds are currently being identified. But one of the more exciting things, as I've mentioned, is that repurposing of FDA-approved drugs is the fastest way that we can get to new therapies. And for that, we're working with NIAID, who is providing to Andy Messaker at Purdue all of the protease inhibitors that are currently FDA approved. And we're screening those with these activity assays that are developed against 3CL-Pro and PL-Pro. And so we're hoping that from this, we can actually identify protease inhibitors that are already FDA approved that can be advanced very quickly to clinical trials. So now if we look at the next step, we've moved from proteolysis, we can follow that next area to looking at the replicase. The replication, so if you think about how translation occurs in eukaryotes, that first open reading frame is going to be translated from the nascent mRNA, but you need to have new mRNAs to make all these other proteins that make up the particle. So the first mRNA you see here is that spike protein, but and the E protein and the M protein also part of that particle. There's also other small proteins that need to be made. Many of these are involved in immune evasion. And so we want to be able to make sure that this virus is moving forward without actually being detected. So the replicase, the structure of this has also been solved. This was released from a lab in Beijing on March 17th. And we see that this is a large protein built of NSP15 bound to two molecules of NSP8 and one molecule of NSP7. The excitement for this molecule is that also we know that the replicase includes two other proteins, NSP9 replicase and the NSP15 RNase. And so these are not part of this larger structure, but the crystal structure of these were solved by CSGID, and these have also been released to the public. So why is this structure so important? That's because this is the protein that is believed to be the direct target for remdesivir, which we saw from Michael Ison's talk is currently under clinical trials. And that's because it's been shown to be a direct inhibitor of this RNase-dependent RNA polymerase from MERS. And so there's people not only trying to work to build this structure for structural biology, but also to get activity going so remdesivir can be tested. There's also a really great need to actually get this complex and the structure solved with remdesivir bound to it. Because if remdesivir is not the perfect drug, a small modification to it may enhance its activity against COVID-19. Then if we think about the next stage that we can inhibit, once these mRNAs are made, they actually need to go through a capping reaction. And the capping reaction is very important because as this mRNA extends, we don't want it to have it be just simply degraded. And so the cap actually protects the ends of all mRNAs in host cells. And so for the capping reaction, you have two stages. One, you have these two phosphate residues that have to be clipped away, and this 7-methylguanosine cap is added. For the virus, this is actually stimulated by a protein complex of these NSP proteins, NSP10 and NSP14. So this 7-methyl cap is added by those CoV-2 proteins. The second reaction that you need is a 2-O-methyltransferase. And that's actually going to add a methyl group into this other site, which allows this transcript to not be recognized by the immune system. Our immune systems are able to detect if a viral RNA is present, so the methylation at this site protects from that avoidance. So there's structural biology going on here as well. There's not a structure yet at the NSP10-14 complex, but that complex is 95% identical to that from SARS, and so there's drug discovery going on already just based on its homology to SARS. That structure was published in 2015. You see here the NSP10 in red, covalently bound to the NSP14, shown in green and blue. This is bound to that methyl cap, GPPA, as well as to the byproduct of the reaction, which is SAH. Here at Northwestern, we actually looked at the other side of this reaction. We recently released the structure of NSP10 bound to the other methyltransferase, which is NSP16, and this was bound to the substrate molecule S-adenosylmethionine. And again, this is a really a great target for drug discovery, because if you inhibit it, you would actually get increased detection of the virus by TLR7 and TLR8, which could lead to better detection of the virus earlier on. And so what we're doing now at Northwestern specifically is we're moving this through partnerships with industry into looking for peptide mimics that can block interactions, inhibitors that block the binding site for the S-adenomethionine, methionine, as well as looking for novel inhibitors. But this is really getting into that zone, that slow zone, as de novo discovery as opposed to repurposing, because there was no drug development done against NSP, against the methyltransferases for SARS. So finally, I just want to summarize that there's a worldwide effort to use structural biology for drug discovery. I've given you the examples of what's publicly available now, but there's certainly other sites that are going. I presented work from our center as well as our sister center in Seattle. Jihei Rao's lab at Sinseng University in Beijing has been actively studying this over the past 5 years, as well as the Diamond Light Source in the United Kingdom. And I just want to finally talk about where the next steps will go in drug discovery. We now have a large pool of proteins that have been built and developed. This allows us to do high throughput activity screening. First off, the most exciting would be to repurpose FDA-approved drugs that inhibit these proteins, but also retesting lead compounds that were identified in early drug discovery efforts for SARS that were then abandoned, rebooting those efforts. But also, I think we need to be looking at identification of novel lead compounds, fragment screening, and ligand screens that would be effective in the future for treatment of these. We're going to continue structural biology here at Northwestern and throughout the consortium of putative targets. Again, NSP14 is a target that's still going, as well as improving structures that we've already identified to get those that are less than 2 angstroms so we can get improved drug discovery. Then you refine those compounds, test lead compounds for antiviral activity, test for in vivo efficacy and pharmacokinetics. Here, a really important aspect is that mouse models for COVID are actually starting to come online. Many of you may know that mice are not susceptible to SARS-CoV-2, but humanized mice have been developed. Breeding programs are going on throughout the world, and those breeding programs now are starting to be successful. And then we move on to clinical trials. So finally, I just want to say that drug discovery can be a slow process, but we are already off the starting blocks, and we actually are on record pace. Gene to structure has happened in a minimal 2-month period of time, and now there's movement now to lead to drug. And so with that, I just want to remind you that I'm speaking on behalf of the large consortium, and I will turn it back over to Robert Murphy, who is the coordinator of the session. Thank you.
Great. Thank you very much, Karla and Mike and Chad. We're a little bit over, but I think we can take a couple questions. They've been coming in. There's a question here. Does recent infection with milder strains of coronavirus have any protective effects at all against COVID-19? And is this perhaps why children have such mild disease? Mike or Chad or anyone want to take that? Mike, how about you?
Sure, yeah. So I'm not sure I would say milder strains. I don't know that we have enough data yet to know strain-to-strain variability in terms of severity of illness and whether the severity of illness has something to do with the virus, the host, or some other factors such as pre-existing antibodies.
How about other coronaviruses, though?
So other coronaviruses do not confer cross-protective neutralizing antibodies. So, seasonal coronavirus doesn't, and other animal coronaviruses should not.
Do we have other questions, Kate? I see it. Okay. Let's see. As far as I know, asymptomatic spread in China was known in early January, and preventive measures were adapted accordingly, but it was not taken into consideration in other parts of the world until recently. Could you comment? I guess that would be Chad.
Yeah, I mean, I think there was some uncertainty, but I think with that information is why we eventually instituted the social distancing measures that we have in place now. I think the best way to really combat asymptomatic transmissions is through that method. And, why or when we got to those recommendations of social distancing is a very complicated question and answer, but we have known that there is some evidence of possibly asymptomatic spread early on, but I don't think that we really had good, established data on that until a little bit more recently, even this week, with the cluster analysis that they were showing from Singapore that was in MMWR this week that showed there was clearly pre-symptomatic transmission. So essentially the people were asymptomatic at the time they transmitted, and they were able to document transition in that fashion.
Another question, do you think we really made a mistake at the beginning by not requiring masks more universally? Brad?
I think that's a tough question as well. I mean, I think masking is something that they learned in Asia after SARS-CoV-1, right? So, and I think that was when masking became a big thing and more acceptable within that society and used more readily. So I think experience helped them. They understood, I think, this notion of social distancing is great, but there are situations where you can't keep that six foot bubble around you. And I think in populated cities in Asia, they understood that upfront better and then just had a more universal masking policy. I think here we felt, I think, because we haven't gone through that before, that maybe we could do the distancing and we could avoid masking potentially. But just being a healthcare worker working around the hospital, you realize pretty quickly that everyone is crowded. It's just the nature of the building and the place and where we congregate for rounds and for doing our work is a crowded environment. So anytime there's that type of crowding that we can't really avoid, I think masks have the potential to be helpful. And then out in the general public as well, there are some situations where you're kind of forced to be somewhat closer to people, and it can be unpredictable when that happens. So I think this is just another barrier of protection.
Karla Satchell, why was SARS drug development halted?
So drug development, unfortunately, is a profit-driven spectrum. And so when SARS went away, there was deemed to be no need for it. When MERS came, there was actually a rebooting of that. So you do see some of these structures and things have been solved since the MERS epidemic. And so there was some advance, but the community is very small. There's not been a lot of research, and there hasn't been a real strong economic driver to have that research forwarded. And so a lot of it was just stopped. It's really crazy to read the literature. You read up to 2009, say, and really it falls into just 1, 2, maybe 3 groups throughout the world.
Mm-hmm. Does anyone know about the durability of the immunity after initial infection, maybe related to SARS and MERS and other coronaviruses? What the prediction is for SARS-CoV-2?
So based on seasonal coronaviruses, MERS and SARS, people typically get pretty high antibody titers that decline over time. We haven't had, for most of the newer SARS and MERS, patients that have gotten re-exposed to know of how protective it is. For seasonal coronaviruses, usually those antibodies wane over a year or two, and that's part of the reason why people can get reinfected every couple of years. So probably will follow that, but we'll have to wait and see.
The described models that were presented, I think mostly by Chad here, but a little bit by Mike too, were largely based on the data from China. Are these data now under any kind of question based on the information that's coming out of the U.S.?
The data from China under question, I guess, is the question. So,
well, I mean, a lot of the models, the information was based on Chinese data or heavily influenced by the Chinese data. But now we're having our own data. We have data from other European countries. Are the models really adapting to what we're seeing here? I think that's the question.
Yeah, I think that's the idea. Once we get that information, those modelers that do those things will change assumptions based on the kind of local geography and timing. But it's the best information we have, I still think, is from the looking at the full epi curve of what happened in China as to where we can get the most information about assumptions regarding the reproductive rate and the transmissibility, the incubation period, all those different parameters. When looking at those different parameters, the incubation period has actually been pretty consistent across different studies in different parts of the world. And so that's one thing that's been fairly consistent. And I think that just that the issue of how transmissible that is so time dependent, and it depends upon the also the strategies, epidemiologic measures that are being put into place to reduce transmission. But hopefully we get more information over time as we have more data that can be locally applied in the United States.
Are there any plans, maybe this is for Karla, any plans now on collecting the data and samples for future research, assuming that SARS-CoV-2 ultimately goes away somewhere? What's the future plans? I hope it's not going to be like a SARS-CoV-1 and everything comes to a halt.
Yeah, I think that what I hear a lot of people interested in as we're looking at developing new drugs for SARS-CoV-2 is checking those against bat SARS and checking them against SARS for hopes that we can get broader applicable. So trying to identify drugs that are more shared across coronaviruses, even getting into those different classes, so that if something emerged in the future, drugs developed now would be applicable in the future. So not getting to these highly variable regions, but to the much more conserved coronavirus region.
That's great. I think we really kind of run out of time. I want to thank everyone, all our panelists, Karla Satchell, Mike Ison, Chad Achenbach. I want to thank everyone for joining in, for their questions. I'm sorry we didn't get to many more. However, I want you to know that the presentations will be on the website, the Global Health website. It also is on our Facebook page and Twitter page, and there will be a Feinberg podcast available next week that's actually even eligible for CME credit. Also, if you want to get on our listserv, we're going to be having other COVID-19 and other emerging infectious diseases discussions over the year. You can sign up on our website and get on the listserv. Also, just of note for those in the research area, the Institute for Global Health has a Catalyzer Award notice out right now with rolling applications being taken. These are for pilot studies related to COVID-19, approximately $25,000 for a one-year project or starter project. The applications are open now, and the guaranteed turnaround review time is 1 week. So I encourage anyone that's interested in a small pilot project like that they can apply now. And finally, our next First Friday presentation is May 1st, so please book that in your calendars now. Same time, noon, it'll be a virtual presentation just like today. So again, thank our panelists for their wonderful presentations, and thank you for participating today.
Thank you so much.